Good morning, and welcome to this week's Columbia Psychiatry Grand Rounds. And before we get started with today's excellent program, I just have a few announcements that I'd like to make. So our next week, Grand Rounds, we're going to have the second in our clinical update series, and that's going to be titled The Therapeutic Use of Psychedelic and Dissociative Medications in Psychiatry. And it will be comprised of three separate talks delivered by Dr. Manit Kishan, who is an associate professor of clinical medical psychology, Joshua Berman, who is an assistant professor of psychiatry, and then Dr. David Harlestein, who's a professor of clinical psychiatry, all from our own Columbia department. I also want to let everybody know that April 20th, we are going to begin our first hybrid rounds with Dr. Suzette Evans, who's professor of clinical psychiatry, also here at Columbia. And the hybrid format will allow for in-person attendance in the auditorium, as well as a simul broadcast via Zoom for those who would prefer to view remotely. So we're very much looking forward to resuming the option of in-person rounds. So please watch out for those announcements as we near that time. So now I'm going to turn it over to Dr. Muskin to introduce the lecture today.
Thank you. Hi, everybody. Good morning. This is one in a series of Spiegel Grand Rounds. As you can see, before you, Herb Spiegel was a faculty member for half a century at Columbia and was one of my important mentors. The first Grand Rounds, the Spiegel Grand Rounds, was actually given by Dr. Spiegel, I think in 2006, followed by his son David Spiegel, both well known in the world of hypnosis. Let me tell you just a little bit about Herb before we start today's Grand Rounds. Herb was trained as a resident at St. Elizabeth's Hospital in Washington and learned hypnosis there. During the Second World War, he was a battalion surgeon in North Africa, and he used hypnosis to return shell-shocked soldiers to the front. And as Herb told it, they used to give Amitol, and one day he realized that the Amitol had not gone into the soldier's arm, but had spilled out on the floor. The IV came out, and yet the person was deeply engrossed in the horrible experience that he'd had that had sent him back to the hospital. And he wrote, "I later discovered that it was possible to use persuasion and suggestion to help the men return to previous levels of function after sustaining severe combat stress." He was, as I said, a clinical professor of psychiatry for many years, which is where I met Herb when he taught postgraduate courses on hypnosis in what was then called the Alumni Auditorium. He really was a pioneer in the use of hypnosis as a tool to help patients control pain, stop smoking, control their eating, shed phobias, and ease anxieties. And he noted that until the late 1930s, hypnosis was really in the domain of what he called quacks, but he gave credit to them because they kept the practice alive. And to quote Herb, "We are in debt to the quacks for keeping it alive until the medical community started to investigate and find out what a useful tool hypnotism is." So he really established hypnosis as a legitimate medical therapy. And the New York Times wrote in the mid-70s that Spiegel was one of the people whose work over the past few decades has helped strip away the aura of charlatanism and make hypnosis a respectable medical tool. And in the 80s, UPI wrote about him, and to quote from that article, "The prevalent and wrong attitude in the practice of medicine is use a pill or scalpel or a gadget for problem solving. Modern medicine puts such extreme emphasis on high technology and drugs that it often overlooks the oldest and at times the most effective therapeutic instrument that humans possess, the mind. Medicine resorts to its last instead of first. Hypnosis, which accomplishes alterations in human awareness, is a great way to very directly and quickly get people to alter pain." So Herb became really a noted advocate of therapeutic hypnosis in the United States, and he actually was quite a celebrity. And the New York Times said, that actors came to him to overcome stage fright, singers to quit their smoking, politicians to overcome fears of flying. And some people might know this, that Herb had a regular table at the restaurant Elaine's, which is sadly gone. And his table was actually near Woody Allen's table, was really a fixture in the New York celebrity community. Herb died in 1999 in December. He was 95. And the most remarkable story, I know this from Marsha Greenleaf, Dr. Marsha Greenleaf, his widow, that day Herb had gone to Lenox Hill Hospital to do a consultation, had seen a patient, they'd come home, had dinner, and he went to bed and died in his sleep, which I think is a gift from the Almighty, to get to do what you love and then go off to sleep and pass on. So as I said, Herb was an important mentor of mine. I trained with Herb and learned hypnosis. He also was always available if you had a question about a case, a hypnotic case. And it is because of Herb that I met Dr. Ochsner, and because of that Dr. Ochsner will be receiving the Spiegel Award for 2022. Thank you.
Thank you. Okay, but before I introduce our speaker, just a quick reminder that we encourage everyone to post questions at any time during the talk in the Q&A feature at the bottom of the Zoom window, which is separate from the chat. Also, if you're a trainee, please put the word trainee at the beginning of your question. And finally, as we've been doing recently, if you would like to ask your question in person, please add "can ask question myself" or "prefer to have my question read" at the end of your question in the Q&A. And if you'd like to ask your question yourself, what I will do is temporarily promote you to host, and you can ask your question directly to Dr. Ochsner, camera on or off as you'd prefer. Asking the question yourself does add to our sense of connection during Zoom sessions, so we do hope that you'll consider it. Okay, now on to today's speaker. I'm very happy to introduce Dr. Kevin Ochsner, who is the professor and chair of the Department of Psychology at Columbia University. Dr. Ochsner received his PhD in psychology from Harvard University, where he completed a postdoc fellowship before completing a second postdoctoral fellowship at Stanford University in functional imaging. He then joined the psychology department at Columbia University in 2004 as an associate professor, where he has remained for the duration, becoming a full professor in 2014 and assuming the role as chair of the Department of Psychology in 2019. He also directs the Social, Cognitive, and Affective Neuroscience Lab, or the SCAN Lab, which studies the behavioral and brain bases of emotion, self-control, and person perception. He is a prolific author of over more than 140 scientific articles across an array of journals, including Brain, American Psychologist, Neuron Science, and the Journal of Cognitive Neuroscience, also the editor of 3 books. He is the recipient of numerous awards, including today's, but also Young Investigator Award from the Cognitive Neuroscience Society, Columbia University's Lenfest Distinguished Faculty Award, and the APA New Investigator Award. His research has been funded for several decades by grants from private and public institutions, including 5 different NIH institutes. Dr. Ochsner was also the co-founder of the Social and Affective Neuroscience Society and is a past president of the Society for Affective Science. Importantly and impressively, work emanating from Dr. Ochsner's SCAN Lab is among the most cited in the fields of social and affective neuroscience. And Dr. Ochsner has been identified by the Web of Science Group as one of the world's most influential cross-field researchers of the past decade, as indicated by multiple highly cited papers that rank in the top 1% of citations for field and year. So today it is my great pleasure to introduce Dr. Ochsner, whose talk today is titled Evolving Perspectives on the Neural Bases of Self-Regulation. Thank you so very much for joining us today.
Well, thank you so much for the introduction and for the introduction to the Spiegel lecture. Let me see if I can share my screen. I hope everyone can see that. So today, what I'm going to hope to do is to share with you what I'm referring to as an evolving perspective on self-control that's informed by brain imaging research. And it's going to feature centrally the study of types of self-control that vary across the spectrum, from being highly elaborative and cognitive to those that might influence without much deliberate effort at all the initial appraisal of a stimulus, including, we think, hypnosis, as you will find out. And in a nod to the recent occurrence of the Oscars, I'd like to begin with the movies. And one of my favorite movie trilogies from the late Polish director, Krzysztof Kieślowski. You may be familiar with this trilogy, Blue, White, and Red, which deal with different emotional themes of grief, rejection, and betrayal. Of these three films, my personal favorite is White, for its portrayal of a sad sack Polish hairdresser who lives in Paris at the start of the film. Unable to consummate his marriage, his wife decides to divorce him. He's left penniless, bereft, and disconsolate, and finds himself contemplating suicide in a Paris metro station. Happily for him, he runs into an old friend from Warsaw, to whom he recounts his sad story. The friend says, "Why don't you go back home? Go back to Warsaw." The problem is Karol is penniless and can't afford this, so they hatch a plan where they'll put Karol in a large trunk and put him, when his friend travels, they'll check it as a bag on the airplane. Amazingly, this crazy plan works. And we see here the suitcase moving on a conveyor belt, leaving the plane en route to the baggage claim. Unfortunately, this fable takes another terrible turn, where it turns out there's a group of thieves working as baggage handlers at the airport. They steal this large trunk, thinking it contains untold valuables, take it to a garbage dump to open it up and discover its treasures, only to find there's a living person inside. Angered and frustrated, they beat Karol, throw him down this hill into the garbage dump, and as Karol lifts his head from the frozen waste, does he despair? Does he express depression, trauma, and so on? No. Instead, he says, "Home at last." And this story is a fable about his journey from an initially traumatic beginning to finding redemption and ultimately positivity for himself and others. And if you study this from a basic science perspective, a lot of research will begin with the premise that the strong emotions elicited by this individual can be adaptively worked with, modified, and changed through various types of self-regulatory processes. And the question motivating my presentation today is, how does this self-regulation of emotion work? And the talk itself will be divided into 4 parts. In the first, I'll explain how we first proposed a model nearly 20 years ago for how this might work, and how I'll give you a quick synopsis, a quick hit summary of ways that it's been tested for 10 to 15 years. I'll transition into the way we've been translating that basic model in important ways. If there is time, this may be a part I have to skip. We'll talk about how that model, the basic model itself, has evolved to include pieces that we just simply left out in the beginning, but have come to appreciate over time could be important. And then finally, I'll consider the big picture and where this work goes in the future. So let's zoom back to think about how we built the model. At the outset, it's important to explain the overall approach. And we begin with the premise that all of science is about connecting descriptions of phenomena at multiple levels of analysis. So we're concerned, for example, with various kinds of social, cognitive, or affective performance that we measure with responses to stimuli, measures of behavior, and laboratory tasks. We want to link that at a level down to our theory of the mind, the information processing systems, the psychological processes and mental representations that give rise to the behaviors and experiences of interest. And in turn, we want to link that to descriptions of the brain systems that give rise to the processes that give rise to the behaviors. And in today's talk, you'll see me make use of behavioral measures of affect and control. You'll see me make use of fMRI measures of activity in cortical and subcortical systems, and we're going to use those to kind of constrain or triangulate on an account of the affective and cognitive processes that underlie self-control. And so when we first wanted to propose a model guided by this approach, we started with psychology, which generally traffics in the behavior and information processing levels of analysis. And we turned to a model of emotion generation and emotion regulation from our good friend and colleague, James Gross at Stanford. And it begins with the premise that emotions, the triggering of an emotion involves a multi-step sequence. You encounter an elicitor of emotion in a particular situation. You attend to certain aspects of it. You appraise those with respect to your goals, wants, or needs, and that generates the various experiential, behavioral, and physiological components of an emotional response. Now, critically, according to the so-called process model of emotion regulation, you can understand different strategies for regulating emotion in terms of the stage of this emotion generation sequence that they impact. For example, strategies that modify your situation or your proximity to a trigger, get away from it, avoid the bar if you're alcohol dependent, right? Avoid the aisle of the grocery store that has the cakes and the candies if you're on a particular kind of diet, and so on. Modifying proximity to stimuli and availability of them is one class of strategies that act on this initial stage of emotion generation. Attentional deployment changes the degree to which you're focally attending to or kind of distracting yourself with other mental activities when in the face of a stimulus. There are various ways in which you can cognitively change the way in which you appraise the meaning of a stimulus. We'll focus on that in a moment. And then finally, you can modulate the response. You can change whether or not you're showing your feelings on your face and your body. You can never let them see you sweat, keep a stiff upper lip, and so on. But for today, we're going to start off by focusing on reappraisal, which in clinical context is meant to model something like the processes that go into reframing, rethinking, cognitive behavioral therapy, and so on. And we're going to develop a model of that. And we're going to combine the psychological analysis with insights from cognitive neuroscience. And at the start of this work, the model had two parts. The first was the idea that there are a whole suite of control-related brain systems that could play important roles when you're trying to reframe the meaning of an event to change its emotional impact. So first, we thought there could be a whole series of lateral and medial, prefrontal and parietal systems that are important for holding in mind your goals, to regulate your emotion. They could hold in mind the content of a strategy. They could control the focus of your attention on strategy relevant aspects of your emotional situation and ignore others, and so on. We thought there could be important participation of a region in your posterior medial and cingulate cortex, highlighted here, that's thought to be important for monitoring the extent to which you're doing what you want to be doing. It gives you a little signal that says maybe there's a discrepancy. Maybe you need to exert control and it's beneficial to do so. Certainly that happens in varieties of forms of control. We thought that could play an important role in emotion regulation as well. And then we also thought ventral and lateral regions of your frontal lobe that are important for selecting a goal relevant action, mental representation or thought, and inhibiting irrelevant ones. Stroop task, for example, name the color of a word, ignore what you're reading. Ventral outer prefrontal cortex, very important for selecting that color naming response. We thought that's important when you're reappraising. You're selecting among possible ways of thinking about what's happening to you, the intentions of other people, what's going on. And speaking of the intentions of other people, we thought dorsal medial regions that are among the most reliable findings in all of social and affective neuroscience. This region is active anytime you're thinking about the goals and beliefs behind actions. People did it because they wanted something. They had a goal. They're gregarious, they're friendly, they're nice, they're competitive, they're cooperative, and so on. Attributing those mental states, rethinking what they are for yourself and other people could be very important for reframing the meaning of what's happening to you. So if those are the suite of possible control systems that could play a key role, what about the emotion systems they might impact? Well, I'll highlight just a couple for present purposes. The first in this transparent view of subcortical systems, as if you're looking through a transparent view of the kind of frontal lobe, or sorry, my left. You can see two regions highlighted here with these kind of pink circles, the amygdala, which means almond in Greek, almond-shaped incised cluster of nuclei that seems to be extremely important for detecting stimuli relevant to your goals. And especially those that are of potential threat value, helping trigger appropriate responses. And then cheek and jowl with that, there's another system known as the ventral, or the bottom part of your striatum, which just looks striped in a cross section of the brain, as you know. That seems to be important for encoding the reward value of stimuli. And we thought these could play different types of roles when you're experiencing and then trying to modulate aversive and appetitive affective states. Okay, so that's where we started 20 years ago now. Incredible for me to believe. And let me just quickly give you the capsule, a 10,000-foot summary of 15 or so years of work from my lab and other labs around the world that have tested and elaborated this model. We're going to highlight a question and give just examples of citation. So the first question, I've already alluded to what the answer might be, is as you're regulating qualitatively different types of emotions, say positive versus negative, broadly construed, what systems do you see? Well, if you're modulating negative, aversive responses, on average, you're going to see amygdala involvement. But if you're regulating appetitive responses, such as craving for food or alcohol or cigarettes, if you're nicotine dependent, you'll see modulation of ventral regions of your striatum, as if you're modulating the expected reward you might have if you consume these substances. We also have tested the linkage between control and affect systems to change emotional experience. And we found, for example, that activation, recruitment to prefrontal regions leads to a drop in negative affect, where this is an abbreviation for looking at and responding to a visual stimulus naturally. And this is an abbreviation for trying to reappraise it. And this downward pointing arrow is the drop in self-reported affective experience that you experience that happens as a result of reappraisal. So the magnitude of that drop is predicted by prefrontal activity, but importantly, it's mediated by changes in activity in these subcortical systems, like the striatum and the amygdala, which is what you'd expect if this model is correct. What about qualitatively different types of strategies? I've focused on reappraisal, but what about when you're just trying to distract yourself? Like try to think of something different, take your mind off the worries and troubles of your day. Well, reappraisal seems to depend more on these medial and ventrolateral systems that we think are important for thinking about what a situation means and selecting a context appropriate meaning, whereas distraction seems to depend more on these dorsolateral, prefrontal, and parietal control systems that we already knew were important for selectively attending to one type of stimulus or another. In the case of our experiments and those of others, it's trying to attend internally on something like that internal thought, find your happy place, find an alternative mental place to go, and ignore the aversive things going on around you. We've also looked at different varieties of reappraisal. So I've focused on one that we call as a tactic reinterpretation. That's the telling a story. You know, that person didn't mean to make that offhand comment in a mean-spirited way. They really thought it was going to be funny, so maybe I can forgive them, for example. And we've contrasted that with distancing, which involves trying to be psychologically separate, be objective, try to view what's happening as if you're a fly on the wall from a video camera perspective. That reliably depends more on brain systems for self-awareness and controlling the focus of attention relative to reappraisal, which depends more, reinterpretation, which depends more on these frontal systems for selecting context-appropriate meanings. We've looked at other types of ways to sort of reframe, looking on the bright side and finding positive meaning, or just pretending it's not so bad. When you minimize the negativity, you turn off the amygdala. When you're finding positive meaning, you not only engender activity in medial and striatal systems important for reward, you enhance functional connectivity between these regions and other control systems. And then finally, we've looked at how the timing of trying to regulate matters. You can show, for example, that if you anticipate the need to regulate, so maybe you're visiting a sick loved one in the hospital and you're trying to practice ahead of time. This is going to be hard. How should I think about it? Those kinds of pre-appraisal settings show anticipatory activity in frontal systems for perhaps selecting possible ways to reframe. And then later on, when that event happens, what you find is that amygdala decreases are the mediator of that prefrontal activity actually changing your emotional response. And then finally, we've asked, what happens if you repeatedly try again and again to reframe the meaning of something that's particularly upsetting? You had a breakup with a loved one, with a romantic partner. Later on, weeks later, you run into them again. Is the flame still flickering in the background, or has it gone out due to your repeated attempts to sort of find the reason this was the right thing to do, that you broke up with them? What we find is that you can see even a week or longer later in recent work, amygdala decreases due to reappraisal will last. They will endure if you've repeatedly tried to kind of reframe that one negative event. Okay, so that's a quick whistle-stop tour of about 15 years of research across various labs to kind of propose and test an initial model of self-control and self-regulation as applied to changing our emotional responses. Let me now turn to the bulk of what I'm going to tell you about today, which is how we've taken this model and attempted to translate it to clinically relevant contexts. And to do that, we're going to take this approach and we're going to translate it across levels of analysis. One way to do that is to move from our lab-based measures into asking how our model of the self-regulation of emotion can speak to everyday behaviors as assessed, for example, using ecological momentary assessment, so pinging and asking people questions at random intervals in their daily life. We're also going to ask how we can connect up to person-level descriptors, groups with different clinical diagnoses, people of different personality or trait characteristics, and so on. And we're also going to start focusing on a different type of self-control known as mindfulness. But before we get to that, let me just give you a couple of specific examples of how this model, the basic model I already laid out, has been somewhat successful in translating to clinical domains. So if you take this basic model and you want to ask, for example, can you characterize dysfunction in either control or affect systems for, say, patients with borderline personality disorder or major depressive disorder? Those are the two that have been looked at the most. Here are some example citations. You'll find, for example, in many studies, you'll see either stronger responses in these patient populations in the amygdala to aversive stimuli, broadly construed. You might see that the amygdala has altered connectivity in patients as compared to healthy and typically developing groups. And in some cases, the nature of that connectivity or the magnitude of activity there may predict the severity of depressive or borderline symptoms. On the other hand, some studies also find altered or weakened activity in control systems, such as your ventrolateral prefrontal cortex or cingulate cortex, suggesting that this model is capable of starting to provide an account at the neural level of the mechanisms that may show altered function in certain clinical populations. And just to give you one example from a collaboration with Barbara Stanley at the Columbia Medical School, you may know Barbara. We were interested in borderline personality disorder, which you may know is characterized by two types of primary problems with emotion and emotion regulation. On the one hand, there's great affective lability. I think of this in a cartoon-like way as affective triggers kind of move at a more frictionless surface. They elicit just a greater magnitude of response. And as you move from one trigger to another, those responses can ping out. On the other hand, they report difficulties regulating their emotion. These two sort of facets of BPD can be measured using standardized questionnaires. In this study, we did that. These are citations for those questionnaires. And we were wondering, could you see the degree to which you show affective lability predict, say, amygdala response, whereas the degree to which you report having difficulties regulation predict a failure to engage prefrontal control systems. What I'm going to show you here are the results that we reported in this paper, where on the y-axis, it's activity in the amygdala or activity in a region of ventrolateral prefrontal cortex as a function of your score on a measure of affective lability or a score on the difficulties in emotion regulation scale. And the simple story is the correlations are exactly as you would predict. If this model and these questionnaires at the person level of description are picking up the psychological operations that these brain systems support, and that in turn might be dysfunctional in this patient population. Okay, so far so good. But what I want to do now is pivot to thinking about a different way of thinking about self-control. If reappraisal is sort of an active Western, get in there, rework it, reshape, remold, alter the way you think about a situation, mindfulness is the flip side. It's characterized by attention to the present moment, but has a distanced, accepting attitude that allows emotional responses to rise and fall without judgment. It's become a core feature of various types of interventions to reduce stress, substance use, alcohol use, and especially cases where the trigger and the problematic resulting affective states are chronic and are not easily susceptible to other types of regulatory strategies. You have to live with chronic pain. You have to live with an addiction. You have to live with some type of traumatic experience and so on. How do you do that? So we wanted to start using functional imaging as a tool to understand how it works. And on the one hand, you might think, okay, maybe it's going to work the same way as reappraisal or distraction. These sort of intensive, cognitive, top-down strategies that recruit these lateral and posterior medial frontal systems. However, there's an alternative possibility here, is that it works from the bottom up without effortful top-down self-control. Here, the hypothesis is that it might fundamentally change your goals when encountering stimuli. You might no longer be looking for and fearful about the consequences of experiencing potential threats, whether that's a chronic pain syndrome or what have you. And your emotions then may change because emotions arise from the appraisals of the relevance of stimuli to your goals, your wants, and your needs. And if those goals fundamentally change, then your emotions might change as well. So to provide an initial test of this, we collaborated with the lab of Hedy Kober, who's now at Yale Psychiatry, and Tor Wager, who once was at Columbia and is now at Dartmouth. We wanted to compare, in a brain imaging context, situations where you mindfully accept and allow emotional responses to a situation where you just let yourself react naturally. And we trained participants to do this. They were all meditation and mindfulness naive participants, come in off the street. We give them 20 minutes of training on how to do this, where the key for accepting is to notice and accept the emotional response without judgment. You can notice that it rises and falls, but there's no need to add cognitive insult to perceived injury. No need to elaborate, notice and accept, as it happens. The experiment was set up in a series of blocks where they would alternate between blocks where they maintained the accepting mindset or the react mindset. At the beginning of each block, they would receive one of these two instruction cues. And then there would be a series of trials within each block where each trial had a fairly long structure. I'm just going to hit the highlights here. You wouldn't know whether or not at any given moment in time, you would experience or be presented with either an image that is negative or neutral based on normative ratings, or we had a thermode attached to your forearm that could heat up to an idiosyncratically painful temperature for you. Pre-testing established it as painful for you, or just warm as a control comparison condition. And then after experiencing either the images or the heat stimuli from the accepting or the reacting naturally mindset, you'd rate how you felt. And that's going to be a behavioral measure of the efficacy or the extent to which acceptance might change your emotional response. What I'm going to show you now are for the two types of stimuli, behaviorally what happens. On the y-axis is self-reported strength of negative affect in the scanner environment as a function of, for the images, whether they were negative or neutral, and for the heat stimuli, whether it was hot or warm. React trials are going to be in the red bars, and accept trials are going to be in the blue bars, or accept versus react blocks, I should say. First thing is for the images to note, on average, self-reports of negative emotion are, of course, greater on average. That's this main effect here. When you're looking at an aversive as compared to a neutral photograph, but accepting and allowing your responses leads to less overall negative affect. It's a very reliable effect. Exactly parallel effects in the context of heat stimuli. The critical one here is for painfully hot stimuli. When you accept and allow whatever affective response that you have to the, feels like a scalding hot cup of coffee on your forearm, the response diminishes in magnitude. Okay, so far so good. This is proof, at least from a behavioral perspective, that this mindful acceptance is diminishing your response. What's going on neurally? Well, let me just show you two results. The first is if you home in on the amygdala, which we know from dozens and dozens of studies will reliably respond more strongly to stimuli that signal that depict, I should say, potential threats and aversive objects, insects, animals, situations, and so on. Shown here is a time course of activity. It's activity on the y-axis across time. First for the period where the image is on the screen, and then afterwards. And what you can see is that the accepting mindset it's not as if there isn't an increase in response, it just isn't as great as when you're in the reactive and a respond naturally mindset. On the pain side, there's a whole suite of systems known as the pain matrix that reliably becomes active when you experience pain, physical pain. I'm highlighting activity in one of them, portions of the ventral anterior insula right here, that show a very parallel pattern. So that when you're in the react naturally mindset, there's a sustained peak of activity. You'd see very parallel results in the thalamus, the dorsal cingulate, and so on. But that rise isn't as great in the accepting mindset. Okay, so it looks like they're having predictable effects on behavior and the neural systems for responding. But notably, unlike reappraisal or other cognitive forms of regulation, mindfulness did not show any prefrontal involvement in any of multiple types of common analyses, with whole brain contrasts, ROI-based analyses, looking for specifically reappraisal-related regions, and so on. We looked for brain behavior correlations. Nothing obvious turned out. And we were left to conclude that mindfulness seems to have a holistic impact on your appraisal of a stimulus. It could change your goals, so things are just not perceived of as threatening at baseline, but it may also keep you from adding what the Buddhists call the second arrow. The first arrow of injury from a negative event is the event itself, but the second arrow is the fear you have because you're in pain, and then the anxiety you have because you're afraid because you're in pain, and so on. We may not cognitively elaborate these types of stimuli. This got us to thinking that this type of strategy, mindfulness, could be pretty useful in the harried aspects of daily life where we're constantly distracted. Lots of people don't have all their prefrontal resources available to them at every moment of the day to notice and monitor the need to regulate, and so on. So this leads to a second study, which is done in collaboration with folks at Penn and Dartmouth. And I would single out here, especially Emily Falk, who is the leader of this large multi-site project that whose goal is to understand the link between a consequential everyday health behavior that's prevalent for college students and in the military, which funded this research. Drinking and binge drinking can be highly problematic for reasons I don't think I would need to go into for a clinical audience such as yourselves. But I wanted to ask, to what extent could we combine fMRI measures of the use of mindfulness to reduce craving or desire to consume alcohol as triggered by stimuli, such as images of people drinking, and combine that with everyday measures of behavior using ecological momentary assessment, where we would measure people's desires to drink, how many drinks they would have, and related emotions. But we would also ask to what extent we can use mindfulness as an intervention. We give people reminders to do that. Here's the overall layout of the study. Behavioral data was collected at 2 sites among undergraduates at Penn and undergraduates at Columbia. They were randomly assigned to one of 3 groups. One would do a mindfulness-related task in the scanner and would get mindfulness-related reminders in their daily life. There's a perspective-taking manipulation we don't need to go into here. And then there was a control group that got none of this. They got no mention of mindfulness, no mention of any regulation whatsoever, and they got no reminders in their daily life. In the scanner, they would do a cue-induced craving task where they would see photographic images related to consumption of alcohol, including with college-type peers, rate how much they would want to drink, how much they were craving drinking after seeing the images. And we did that in a couple of conditions I'll tell you about in a second. One involved just reacting naturally, the other being sort of mindful and accepting of the craving, just like in the study I told you about. And then critically, for almost a month afterwards, they would get reminders of trying to be mindful about their drinking desires. And we also measured how much alcohol they actually consumed. I'll quickly show you the behavioral results as well from a behavioral replication study. This was done actually during COVID, entirely online. Otherwise, it's the same except for the fact that they did the equivalent of being trained in mindfulness in an internet interface instead of coming to the scanner and doing it. So there's no brain data for this participants, obviously. So the first question you might want to ask is, did mindfulness reduce craving in the scanner? We saw it could reduce negative affect and pain experience. How about here? Does it reduce craving? It's presented a different way than I've shown you the data before. But the critical thing to note is that if this on the x-axis is the extent to which you crave drinking, this is the kind of orangey peach-colored bar, or sorry, peach-colored distribution is for when you're in a reactive mindset, and the blue distribution is when you're in the mindful and accepting mindset. The difference in the means here is significant and reliable. It's not an enormous effect size, but it's definitely significant and it's there. So people are craving or thinking they might want to consume alcohol less when they're in the mindful, accepting state. Okay, now you might want to know, did reminders to be mindful change drinking behavior? So here we had a between group manipulation. Some participants got the mindful training and others didn't. But really, for this purpose, I want to focus on a within participants manipulation, where across 4 weeks, 2 weeks were active, where you'd get the reminders, as shown here on the left. If you're around alcohol today, react mindfully, notice, acknowledge, and accept the thoughts and feelings you have. They get that twice a day. But in control weeks, they would just be told to react naturally. So it's trying to do the equivalent of the scanner task in daily life, but splitting it up in separate weeks across a 4-week interval. So let's go to the data. Were participants less likely to drink during active as compared to control weeks? Again, the active weeks are where they're getting the reminders to be mindful. And I'll show you the data for both the scanner sample and the behavior only replication sample. And we'll do, for the mindfulness group, we'll have the within participants comparison of active versus control weeks, but we can also look at the between group difference as well. So the first thing I want to note, where this is the number of drinks, the proportion of occasions on which they reported drinking, is that on average, the mindful groups are drinking less than the control group, but within the mindful groups, on the weeks where they're getting the reminders, I'm sorry, I have this labeled exactly opposite. These are the active weeks where they're reporting drinking less as compared to the control weeks. And in the behavior only replication sample, you see exactly the same thing, or roughly the same thing. The difference between the groups isn't as big, but the within groups comparison, where this is amount of alcohol, proportion of times you drink in the during the weeks that you get the reminders to be mindful as compared to the active weeks. Okay. But now remember, I said earlier, when we were analyzing the data from our mindfulness, first mindfulness imaging experiment, we weren't seeing engagement of a kind of control pattern engaging prefrontal systems. So we took an alternative approach to analyzing these data, which you can think of as kind of predictive modeling or doing a classifier analysis. And it has 3 steps. In the first, you're interested in figuring out the pattern of activity across voxels, which is our unit of measurement for fMRI in the brain. Are there patterns that reliably distinguish between different psychological states? In this case, being mindfully aware or letting yourself react naturally. And so what we want at the end of the day is a neural signature of being more mindful. And the predictive model here is really just a regression weight. It's the weight that you would expect, the prediction your model makes for relative differences in activity across voxels if you're in a mindful state versus letting yourself respond naturally. And now what we're going to do is, having built this model with machine learning and finding it's about 70% accurate, which is actually pretty good for this kind of application, we're now going to ask, can we use the extent to which you as an individual trying to be mindful express that pattern to predict how much you're in the moment craving for alcohol changes? And can we also use it to predict how effective the mindfulness intervention is for you in daily life? Okay, so let's turn to the scanner first. What I'm showing you here is your craving on the y-axis as a function of the extent to which your signature on any given trial looks like it expresses the mindful signature or a reactivity signature. And what you see is this negative slope, so that the more strongly you express a signature reflecting mindfulness, the better able you were, the bigger the drop in craving going down, down, down here, the better you were able to reduce craving when you were trying to be mindful. So that's great, right? So whereas just doing what's called univariate analysis on average is activity in any one brain region predicting efficacy in being mindful and changing emotion. We didn't see that in the first study, but here using a whole brain distributed pattern, we can get that kind of predictive power. But now let's ask about daily life. Can we translate this to predicting everyday behavior? Now what we're going to try to predict as an outcome is the number of drinks that you have during control or active reminder weeks, being reminded to be mindful. And if you just look on average across all participants, the mindfulness signature is sort of trending in the direction you want, but really what you need to do is look at the people that are a standard deviation above the mean in terms of more strongly expressing the signature. And for those individuals, the more strongly they express the signature, that's the blue line, these strong expressors, they show that the mindfulness intervention is more effective. They report drinking less when they're actively being reminded to be mindful, as if it's reinstating this whole brain signature for mindfulness, as compared to the weeks where they're just letting themselves respond naturally. Okay. Now let me turn to the last behavioral or imaging study that I'll get into in detail. And this relates to the work of Herb Spiegel as a clinical hypnotist. And so we became interested in doing the prior work in how altered states of awareness, such as mindfulness, can have a regulatory impact. And for a number of reasons, we got interested in hypnosis as a potential modulator of the efficacy of mindfulness, and perhaps by extension, other forms of control. And as you know, hypnosis can be thought of as a mental state that involves focused attention, an enhanced capacity for response to suggestion. It can be induced by a therapist or by yourself. In fact, some clinicians who use hypnosis say that all forms of hypnosis are self-hypnosis, insofar as the individual, the client, or the patient being hypnotized has to be a willing participant and a believer that they can change from one psychological state into a trance state. This photograph here was actually taken from the obituary for Herb Spiegel in the New York Times, and it depicts him interacting with a client in the 1970s. It's used, I think, as Phil already mentioned, as a treatment for anxiety, trauma, substance use, and a variety of other things. And what we wanted to do was leverage the use of brain imaging to try to understand how it works. It turns out in the last 5 to 10 years, there's been a handful of imaging studies starting to ask related questions. So for example, we already know that if you induce a hypnotic trance outside the scanner, and then you put people in the scanner and you ask, when people are not told to do anything, they're so-called at rest, it alters patterns of functional connectivity among particular brain systems. We also know from a handful of studies that if you, outside the scanner, induce a hypnotic state, plant a post-hypnotic suggestion, take people out of the trance and put them in the scanner, you can observe modest impacts on behavior. Sorry, this is mostly behavioral work. There's been 1 or 2 imaging studies that frankly didn't have designs that really could inform us about very much, including, interestingly enough, a study from Herb Spiegel's son, David Spiegel, in collaboration with Tor Wager. When we started this study, we noticed that no imaging studies had examined 2 questions that had really been of focal interest to us for a very long time, and were of interest to our collaborator, Philip Muskin. So one is, what's the neural signature of the induction itself? We wanted to actually induce people into the hypnotic state while they're in the scanner. That had never been done before. In addition, we wanted to ask how that state being induced impacts emotional responding and the capacity to regulate emotion through mindfulness. And despite the fact that hypnosis had been used clinically to alter affective responses of various kinds, it had never been studied in an imaging context yet when we started this work. And I still think it hasn't. This work has been led by some folks in my lab, Ben Silver and Faustine Corbani. And it has a multi-step procedure that I'll just very quickly illustrate for you. So we start off at the beginning. Oops, sorry, my window for seeing people got all wonky. It begins with an initial online assessment of eligibility for scanning. And then because we started doing screening during COVID, this money came in actually right as COVID was getting underway, unfortunately. So we've been delayed for a long time in collecting these data. And what I'm going to give you at the end of this study is a description of where data collection stands right now. Some kind of preliminary, early, but really, we think, intriguing and exciting results. We want to characterize the extent to which individuals vary in how susceptible that they are to hypnotic induction using something known as the hypnotic induction profile, which Spiegel developed and pioneered, as well as something known as the Harvard Group Scale for Hypnotizability. We don't need to dwell on that for today's purposes. We then randomly assign people to 3 different groups. And you can think of this as a pilot study, a kind of proof of concept, where the ends here, we would ideally want to be at least twice as large to draw really strong conclusions. Participants would be randomly assigned either to a hypnosis group, a relaxation control group, I'll describe in a second, or a natural history control group, but they get no mention of or exposure during the latter portions of the experiment to hypnosis and so on. And sorry, I should mention here, via Zoom, initially we were doing it in person, but ultimately it's over Zoom. Philip Muskin, as a trained clinical hypnotherapist, would assess and hypnotize people over Zoom. And you can do both of these scales, which involve a hypnotic induction and scoring and assessing the extent of response for a participant. You can do it all over Zoom. That's actually one of the methodological contributions of this study, is that we've proved you can do it, which I'm not, maybe that's been done before, but to the best of our knowledge, it hadn't been done. After being randomly assigned, you would get an app guided, we download an app to your phone, a guided 2-week period of practice where in the hypnosis group, you listen to a hypnotic induction script that was recorded by Phil as the hypnotherapist. The relaxation group would listen attentively to a controlled nature recording that was thought to be relaxing, but doesn't include the active ingredients of explicitly trying to put you into a trance state. And the natural history control group just isn't given anything by comparison. Then when you get to the scanner, you do 2 things. You do 2 types of scans at the start. One is a simple resting state scan. What that means is you're left to lie there with your eyes closed, think about whatever you want. That's kind of a baseline measure of how brain regions are communicating when left to your own devices. But then we do another scan of the same duration where it varies by group. If you're in the hypnotic group, you get the induction. You get Phil's recorded hypnotic induction in the scanner. The relaxation group listens to their nature control recording. And the natural history control group is just left to their own devices for another resting state scan. After that, they go and they do a version of the very first mindfulness experiment we did, this time just with neutral or aversive images, and asking people to experience those images in alternating blocks from either a mindful and accepting perspective or a let yourself react naturally perspective. Okay, so keeping an eye on the time. What did we think we might observe coming into this? Well, first, we thought we might see altered connectivity in brain networks for self-awareness, such as a network I haven't talked about much yet, known as the default mode network. This is a suite of regions we know to be involved in mentalizing, memory, and attention that tend to be strongly communicating with each other when you're left to think about whatever you want. It's default in the sense that this pattern of connectivity is observed whenever you don't expressly give people a set of task goals to follow. But it's importantly thought to reflect self-awareness, monitoring of mental states, and so on. And we thought you might see altered connectivity in that network when you're putting people into the hypnotic state. In other words, in the induction as compared to being left at your own devices. And in some ways, I should emphasize, this is a bit of a conservative test because we already know there's a lot of connectivity in this network when people are left to their own devices. But now we're going to try to focus, if you will, or enhance the connectivity in the network with a hypnotic induction. The other possibilities that you can see regions involved in attention and control, some of which overlap with the default mode network, but may not. We're going to focus on the default mode. The other possibility, the other hypothesis we wanted to test is that the induced trance state, perhaps changed connectivity in the default mode network, can endure and would impact subsequent emotional responding and the ability to exert mindful control. Okay, so $64,000 question. What happened? What I'm going to show you, and this is only about a third to halfway into collecting data for each of the groups. I'm going to focus just on the hypnosis versus relaxation groups because they're the most conservative test, because they both involve relaxation. They differ only, we think, or primarily in the active ingredients of putting you explicitly into the trance state. I'm sure Phil could give you a lot more detail on how theoretically to think about the psychological difference between these states. Okay, so we calculated the amount of connectivity or intercommunication among regions in this default mode network during the initial resting state scan. Think about whatever you want, as compared to either the hypnotic induction or relaxation scans. And so I want you to see where the purple line is the hypnosis group and the relaxation control group is in green, is that it looks like the induction is enhancing just a bit, only partway into data collection. It's enhancing connectivity within that default mode network. Okay, so this is a trend in the right direction. Again, not even close to being done with data collection. But now here's the $128,000 question. Could we show that the extent to which hypnosis enhances connectivity in this default mode network during the induction, so bigger numbers mean in greater enhancements of connectivity, could that predict how much your negative affect drops when you're being mindful? The purple line is what I want you to focus on here. The purple line, the answer is yes, it does, which is to us slightly mind-blowing and exciting. And the green line is the relaxation control group. You don't see the same relationship there critically. Again, early days, it's a trend. Don't want to draw any huge conclusions from this, but we think this is very encouraging. Okay, so in the interest of time, I am going to jump over this third part of the talk where at some other time, I'd be happy to talk to you about the way in which we've taken our basic model and elaborated it to include not just the way you implement regulatory strategies, but how prior to that you make decisions, how you have different kinds of beliefs about regulation being useful or important, and how that plays a role. We've also elaborated that to dyadic contexts, where you can think now, not just about me regulating my own emotion, using my frontal lobe and control systems to alter my affect systems, but how that happens between individuals, how I might use my control systems to make decisions to regulate and implement regulatory strategies that change your affect systems. And so let me just skip ahead to the conclusion, which is sort of taking a step back and thinking about the big picture. So we began with this sort of, at the time, hypothetical, but then initially well-supported model of the way in which domain general control systems. These are control systems you'd see for controlling what's in memory, for controlling the focus of attention, for controlling and inhibiting action, and so on. All the stuff on the left, domain general control systems. Could it be that they, when we try to cognitively regulate our emotion, change activity in affect triggering systems like the amygdala, the ventral stratum, and I didn't talk about it, but also the insula? The answer initially for strategies like reappraisal or distraction was certainly yes. But as we start moving into strategies whose psychological constituents may be different, their mechanisms of psychological action may be different, the answer is this model isn't enough. It really, what brain systems are going to provide a signature, if you will, of effective or ineffective regulation are going to depend a lot on exactly how you are regulating. And as we saw with mindfulness, these control systems aren't really engaged. You can see changes in the activity in these affect systems in the absence of any of that. And I'll leave you with another thought from the movies. From one of my favorite movies as a child, Monty Python and the Holy Grail, which you may know is this sort of madcap comedic retelling of the Arthurian legend that searches for the Holy Grail. And although the characters, the Holy Grail kind of represents for research on emotion regulation, like the metaphorical kind of savior, the ultimate goal, the thing that would unlock all of our problems. And I think of that as being analogous to solving an equation that has three variables, where you want to be able to understand how, for a given person in their situation, what's the outcome of using a particular strategy that should lead them to be successful at regulation as compared to ineffective. Being able to specify all the variables in this equation that produce better or worse outcomes is the ultimate goal of the scientific enterprise here. It's a difficult one. In math, trying to solve an equation with three unknowns is, I wouldn't say it's impossible, but it's more difficult than when you only have one or two of them unknown. And for that reason, a lot of basic science research will hold a couple of these constant and vary only the strategy, or they'll hold the situation and the strategy constant and vary only who is attempting to regulate. But in reality, the situation, as you know, is much more complex. People vary on all kinds of dimensions. Situations vary across multiple dimensions. And the way we think about strategies can vary in many ways as well. What we need is a whole lot of research from a whole lot of people on all of these different problems. And I'll leave you with the idea, or just a couple of glimpses of the way my lab is working with labs at the med school and elsewhere to address some subsets of these problems. So just as one example of trying to come up with neural and behavioral signatures of combinations of these variables that lead to vulnerability or resilience. There's a Conte Center grant led by John Mann, but including a whole cast of characters at the med school and the Morningside campus that tries to look at how suicidal ideation and suicidal risk in the context of major depressive disorder relates to responses to particular kinds of aversive cues and the uses of different types of regulatory strategies. At least that's the piece of this puzzle that my lab is trying to unpack among many others that are part of this Conte Center. And there's a lot of people in my lab now that are focusing on the dyadic, the interpersonal way in which regulatory processes play themselves out, and how that differs from what we're trying to regulate for ourselves. Just one insight we've gotten there is that if I try to help you regulate, it ends up making me more likely to regulate for myself, and I feel less depressed as a result. So let me just leave you with that and thank the gigantic cast of characters that's participated in this research, and I look forward to being able to discuss things with you now.
Great, thank you. That was an absolutely wonderful talk. And I just want to turn it over to Blair.
Hi, Blair.
Blair, you're on mute.
Thank you. Hi, Kevin. It is so lovely to see you present and watch you on your Holy Grail to figure out not only the brain systems of emotional regulation, but what I love about it is really bring it to real life. You're not just a brain imager who's studying the brain, but you're really trying to think about what it means for individual people. Now, with great permission, Kevin, can I speak to the audience for a second? Okay. So first of all, you should know that there's a huge range of people in the audience who you brought out: basic scientists, clinical scientists, clinicians from across our unit, people in education and training. And that shows how important and interesting your work is. But I just want to speak to the audience for a second. We're going to try to do something different with Grand Rounds going forward. We want to make the question and answer period as fun and interactive as we can. Some of us lived through the time when question and answer was sort of a Roman forum, and there was a little bit of a gotcha and who could be the smartest person in the room and humiliate the speaker the most. We're not interested in that. We're not interested in that at all. Yes, ask smart and intelligent questions, but let's have a fun, interactive dialogue and shift that culture. That's change one. Change two is, they're very nice. I'm your chair now. They asked me first, do I want to have a question? Actually, I'm going to go last. So I don't want to be the person who asks the first question. In fact, what we want is for, the goal is for diverse voices across our faculty to ask questions. And we're always going to start by seeing if there's a trainee out there who wants to go first, so we can raise up that voice. I think today our residents are not, they're at a different Grand Rounds. So we may not be able to ask that, but that's goal one. And the second goal is that we actually really elicit questions from across our faculty, and that means basic scientists to clinical scientists and clinicians to educators and trainees. And you're going to see our Grand Rounds organizers actually reach out to some of you when you're on the call to see if you want to ask a question, to try to, again, hear diverse questions. So that's what I wanted to say. And now I'm going to hand it back to Kate. But thank you, Kevin. What a great talk.
Thanks, Blair.
So our first question is going to come from Dr. Randy Auerbach, who has noted that he wants to ask the question himself. There he is.
I'm unfortunately not junior anymore, but I was. It's a pleasure to ask you a question. I just want to start by saying, how impressive the scope of your work and just kind of the breadth of what you're doing. I mean, you encapsulated, it sounded like 15 to 20 years of your life in a very seamless and organized way. And it really is artful in terms of how you presented it. So thank you for sharing it. I have kind of two broad questions. And I think I was kind of thinking about the first part of your talk. And my question was really about how when you're focusing on mindfulness, you covered a lot. And I was thinking how nicely the DMN work that's come out more recently would fit in with that scope in terms of, reappraisal, particularly around the mindfulness work. There's some really nice work in real-time neural feedback in terms of modulating the DMN. And as you noted, the self-reference work within the DMN is also closely tied to rumination, which is very closely tied to reappraisal. And so I guess I was curious, on the one hand, within that earlier body of work on mindfulness, had you probed kind of the DMN at that stage? And then a separate but related question, you showed these really elegant findings in terms of modulating core control networks. And I was curious to see if there was kind of long-term sustainability, meaning like, had you scanned them afterwards to see if those effects actually held over time? And the reason being is there's some recent real-time neural feedback work that shows actually enhanced effects within specific systems, actually, within probably a month or two after kind of the intervention, so to speak, within regions that were targeted. So I know that was kind of a big mouthful of a question, but you can pick or choose what you want to answer from it.
Yes, those are all really exciting and interesting questions. So maybe if I have enough working memory to sort of hang on to all those pieces, maybe we can try to touch on them in turn, and I'll count on you to help me out. So with respect to the default mode network, in maybe the first part of the talk, which was all on sort of reappraisal and to a much lesser extent distraction, sort of paradigm cases of the ways in which we actively online try to mentally rework, in the case of reappraisal, our the attribution of meaning for a stimulus. What does this mean to me? I'm going to tell a different story about that, come up with a different narrative. We never saw default mode stuff for that, and you're looking at it in different ways. It was always, there are certain findings that are just sort of freakily reliable, right? Like I used to, in talks, show the results from, there's now been 3 meta-analyses of reappraisal studies. We did the first one in 2014, but there's at least 2 more there's over 100 reappraisal studies. Every study shows the meta-analysis pattern. It's weird. You just get it. But we haven't really seen it, right? And this was a question. I got interested in meditation. I've had meditators in my lab, mindfulness-based stress reduction, things a lot of people in my lab have tried over times. I've been to meditation retreats, not nearly as much as some former lab members like Hetty, but we really became convinced that if I would have had time to kind of talk about how we've evolved in our thinking, reappraisal and all that stuff is just the tip of the iceberg. And I have an image of like the tip of the iceberg. There's so much other stuff that we haven't even touched with even a decade or 15 years of work. So how do you transition away from that? One is you start studying other strategies that probably work in a fundamentally different way, right? Things that involve reworking a network of brain systems that are important for monitoring what's relevant to you and kind of keeping, I almost think of the default mode network as like a motivational working memory, where a lot of those systems are important for affective learning and memory, for kind of monitoring goal relevance. Like self-relevance is just a version of goal relevance, perhaps the most primal one, because everything is, if the goal only matters if it's yours, right? And so we started thinking, well, maybe states like mindfulness or more recently hypnosis, what they're doing is they're acting directly on these networks, like the default mode network, that assess that. So you'll change the appraisal from the bottom up rather than trying to rework things and like change it from the top down. That was the motivation there. I would love to talk to you more about the real-time neurofeedback work you're talking about. It's something that we had thought about branching into in years past, but there isn't world enough in time to do all the things that we might want to do. And thank God research is done by multiple people in multiple places all at the same time. Otherwise, none of us would ever get anything done, right? It's just too time consuming. And then I've lost what the second part of the question was.
You showed, I guess you showed kind of these elegant findings, which I think were kind of these cross-sectional snapshots, meaning just kind of like just one time in the scanner. But I was curious to see if you see these sustained effects post-scan, just given what we're seeing in kind of the real-time neurofeedback world.
Yeah, so I would love to compare notes on that. We've done a handful of behavioral and one imaging study on sort of durability effects, where the way they've been set up is we usually give you a bunch of what a learning and memory researcher would call massed practice. It's like a bunch of practice all at once, either on one day or spread across 2 days with an overnight memory consolidation interval. And then we'll scan you during the training on day one. We'll scan you during the repetition of training on day 2, but we'll start repeating stimuli, just pinging them at you in the absence of a regulatory instruction to see if your initial uninstructed response to the stimulus changes. And then we'll go back a week later and do that same kind of just representation. And what you find is that there's like a dose dependency. If you just reappraise things once, and this may be exactly what you see with the real-time feedback, you reappraise things once, you see an effect at the end of the scan, like 20 minutes later, it's still there. A day later, it's not. It's definitely not there after a week. If you reappraise, like say 5 times, you'll see it immediately, a day later, and a week later, right? We've thought about Leila Davachi, who's a memory researcher, learning and memory researcher. We at one point had planned out an entire R01 that was all about, let's figure it out. We'll do all kinds of things. We'll look at consolidation effects and so on. Never finished the grant, among other things. But I think it's an exciting and important area of research. So that's as much as we know. And we've done behavioral stuff where whether or not you see practice effects depends specifically on how you train people. So for example, if you're training people to do an idiosyncratic, stimulus-specific, tell a story reinterpretation, you don't get much durability because it's so idiosyncratic. But if you try to go for a more global mindset change, like mindfulness, for example, or just getting people to be more distant and objective and in third person, you not only see that enduring, you start to see, if you do EMA in daily life, you start to see them reporting less stress in daily life. It's as if what you're doing is you're entraining the more the more spontaneous adoption of a mindset that's kind of stimulus general rather than stimulus specific. So
thank you so much. Again, really great talk.
Thank you.
Randy, actually relatedly, this is a question from William Tucker, wants to note, can we speak of a talent for mindfulness, analogous to what Herb described to us as residents decades ago, as a talent for hypnotizability?
Oh, wow. Phil and I have talked about hypnotizability, and I've talked to another kind of trained clinical hypnotherapist about this as well. It's turned out that our attempts to assess people as differing in hypnotic susceptibility, it's not so easy to do, because in the hands of a trained hypnotherapist who kind of titrates and tailors aspects of the induction, they can get a lot of people into a trance state. You can see differences. I might ask Phil to speak to that more so than me. I'm not the trained person in hypnotism. I'm trained in the other stuff.
Phil, did you want to? I know you have a question also.
So it actually, Kevin really touched exactly on it. Herb made it clear that everyone can benefit from hypnosis. And across the bell curve of low hypnotizability to people who, I wouldn't call it a talent, I actually think it's a gift. to have that ability to rework the default mode network and turn on things that are not usually turned on or connected, which is the way I look at hypnosis, and I think we're starting to see that. But it's a state, and what you do with the state is what matters. And he was very clear about that. Hypnosis is not a therapy, it's a state. And then it depends on what you do with it. So part of what I'm really encouraged by and absolutely fascinated to see the data is I saw a lot of people who are highly hypnotizable. We attracted the highly hypnotizable people in New York because it was a hypnosis study. And nonetheless, people were not linear. Some people, I think that was the Zoom quality to it, that they had some trouble. But then I saw people 2 weeks later, a week or so, 2 weeks before they went to the scanner, and everyone loved it and they enjoyed it. That is, they were using the state. So is there a talent for mindfulness? Mindfulness is work and you have to practice at it. And if you practice it, you will get a benefit from mindfulness. There's no question. I would say people who have that ability to dissociate, as we talked about last week with Call, that those people, they have a different brain and they can turn those pieces of their brain on or off or connect them. And I think that's part of what we're seeing now. The suggestion, what's different for the people who've trained with Herb, who train with me, is I changed the trance state, and we agreed upon this, to say to people, open your mind up to new ideas. Take them into yourself. Don't judge them. Now, it's kind of a mindfulness paradigm. I have never done that with anybody before in the almost 50 years I've been doing hypnosis. So I think what you do with the instructions and then what you do with the state is part of what we're seeing. And to go to Bill's point, everyone can practice mindfulness, but not everyone will get the same benefit from it. And that, I think, depends upon your brain, which is also your character pathology, if you have character pathology, et cetera.
Yeah, if I could, thanks, Phil. I would like to build on that. So one of the quirks of being a Columbia faculty member is that if you live in a Columbia faculty apartment, some other Columbia faculty member may have lived there before you. And the apartment I'm giving this talk from our apartment, and the previous resident was a theologian named Robert Thurman. I don't know if you know who Robert Thurman is. He was one of the first Westerners to study with the Dalai Lama, and his daughter, oh, by the way, is Uma Thurman, the actress. And when we moved in here, we kept trying to figure out which was Uma's bedroom. It was very fun. We think it was the one that was pink, but Buddhists like pink. You can't tell. At any rate, so I've talked to Bob Thurman a few times about mindfulness and been led into a mindfulness induction by Bob. And one of the things he talks about is that mindfulness, just as Phil said, it's a skill that you can build. But for him, one of the important things is that there has to be kind of the reason you're doing it. And that in the hands, if you've got the wrong goals and kind of ethics and mores guiding you to doing it, it's not going to be as effective. And that's part of the reason they practice not just mindful awareness, but things like loving kindness meditation, which involves a lot of the same techniques of focused attention and an altered state of awareness and monitoring of breathing, but specifically with a focus on the promotion of pro-social and altruistic motives with respect to others. And I'll just offer that as an aside.
Interesting. I want to switch gears a little bit. We still have some questions about mindfulness, but I want to make sure that we run the gamut here. So this is a question from Meg Heaney. She notes a very interesting talk regarding the alcohol studies. I couldn't quite decipher how much people were drinking alcohol, but it didn't look like a lot. Do you think that your findings would have been even more robust with a sample of regular drinkers?
So that's a great question. So this is the data I presented today are from what you might think of as social drinkers, right? So it's college undergraduates who are self-reporting on the amount of drinking that they have, and the amount of drinks that they might report as an individual on a weekly basis is anywhere from 0 to about 7, let's say, 0 to 6 or 7. Probably they're actually doing more and they're underreporting. Like, we're aware of that fact. And so we thought that might kind of constrain the extent to which we can draw inferences. I didn't mention it yet, but we've had a grant from NIAAA with Nazar Naqvi, who's at CUMC, and John Morgenstern, who used to be at CUMC, and he's now at Northwell. We've had a grant for a few years to use the alcohol cue-induced craving paradigm, exactly the same one that we have here. It's a test bed to study problem drinkers as compared to social drinkers. And we have some behavioral work, a published behavioral study that Nasser is the first author on, that shows that problem drinkers are less able to use kind of a distancing version of reappraisal to downregulate, not just craving for alcohol, but craving for food as a control stimulus. It's like a generalized deficit in downregulation. And we've got we've been collecting imaging data and EMA data from them to do something really parallel to what I showed you here for college population. We're doing something really parallel in problem drinkers and social drinkers as a comparison. So stay tuned. I hope to be able to say more about that in the future.
Great. So then there's a question from an anonymous attendee. There's actually two that are related. So I'm going to see if I can combine them. This person really enjoyed the talk. Not familiar with the background here, but does the hypothesis that hypnosis induction results in an increase in DMN connectivity, where does that hypothesis come from in the first place? And are there other hypotheses that you can envision? And then relatedly to this, if you can hold this next question, how do the changes in DMN activity under hypnosis compare and differ to changes seen in DMN activity under psychedelics like psilocybin?
Oh, that's interesting. Okay, so first question, where does the hypothesis that the default mode network could be involved come from? So the early work on the default mode network came out of WashU, and where they would observe that when we're giving you a visual task, a cold cognitive control task, an auditory task, and they would include what they thought was just a baseline condition where the fixation cross, you're just looking at it. And they would ask, what's active during that fixation baseline condition as compared to the task that's cognitive, linguistic, visual, auditory. All of a sudden, there's this whole network of regions that never showed up in these other tasks. So they called it default just by default because they didn't know what else to call it. It turns out, however, when you look at just patterns of resting connectivity between brain regions, like they're always active. And if you do an analysis of what we think from careful experimental work, our functions associated with each of the individual regions, you see ventromedial regions involved in calculating probably the subjective affective value of stimuli. You see parts of the medial temporal lobe involved in encoding and binding together mental representations, and so on. And so we thought, if a hypnotic trance state is in some ways changing your awareness of the self and changing your focus of attention, kind of broadly construed, maybe one possible thing it could do is to increase communication among these systems that are monitoring what's important to you and your goals. Mindfulness, it's a similar hypothesis. It's actually partly why we wanted to use mindfulness as the type of kind of control strategy that hypnosis could interact with, is because there was some evidence that mindfulness might work in part by affecting that as well. But that's not the only possibility. You could think of there being kind of more classic dorsal attention kind of networks that are often studied in the context of visual spatial attention to stimuli out in the world that involve parietal and dorsolateral prefrontal cortex. We just haven't gotten there yet. Like the data is only partly in, and the analyses I presented today were literally in my inbox yesterday at 3 o'clock. So, because it's like, we normally are not wanting to peek at the data in great detail. And when I first received the gracious invitation to give this talk, we had hoped that COVID wouldn't have limited data collection quite as severely as it proved to. So we had hoped the data collection would have been done actually as of December, and instead we're about halfway through. So stay tuned. We're going to look at other hypotheses as well. These are just the ones that we wanted to start with because they seem to be most. The other thing is we thought if the resting state is the comparison condition, we want to know if what you're already seeing there kind of gets enhanced or changed with the hypnotic state.
Wonderful. So we have Rachel Marsh, who is going to squeeze in one last question before we finish today.
Hey, Kevin, great to see you and wonderful talk as always. You're such an amazing storyteller. I've always enjoyed listening to you. So I was just curious, Have you been thinking about, or have you started collaborating with anyone to do this with children? Because as you know, I study pediatric OCD and anxiety, and we definitely see altered connectivity across task control and default mode networks in them that kind of go away or at least predict response to CBT, which is based on exposures, which I think this reappraisal thing might have something to do with. Currently we're writing a clinical trial with Kate Fitzgerald to try to enhance the functioning of task control with cognitive control training for little kids to see if they respond better for CBT. But one thing, as a yogi myself, I've been incredibly interested in is trying to see how meditation moves these patterns of connectivity, or hypnosis. So it's something, if you're interested in, I'd love to talk to you about, because we have these kids coming in, and if we can try to move the needle a little bit to increase response to CBT, or to prevent full-blown disorder in little kids with subclinical symptoms with meditation, right?
Well, yeah, that sounds super interesting on many fronts, and I very much look forward to a chance to talk about that.
Yeah.
The one thing I can say quickly is in years past, we had a grant with my late colleague and friend, Walter Mischel of delay of gratification fame, and BJ Casey, who at the time was at Weill Cornell, went to Yale and is coming back to Barnard this summer. You may already know that. We had a grant to do studies from age 6 to college age of the basic reappraisal paradigm. And there's a former student in my lab, Jennifer Silvers, who was the lead person for that. And so we have a half a dozen or more papers on that, where it's like, it's not exactly a simple story. It's not as simple as, oh, kids have impaired prefrontal cortex. Like, not really. It's not exactly that. It really depends on the motivational relevance of the stimulus. It's an interaction always. Like, it's always that 3-part equation, right?
Mm-hmm.
It's no simple stories in science, unfortunately, as we all know, as much as we might like them to be simple. So we have a lot of that work. Jen has continued some of that work. And we've had an interest in going back. I've dabbled in aging research recently, and then I decided, nah, for right reason, let's go back to doing developmental work. That's of much greater interest, at least for me personally and to people in the lab. So long story short, would love to talk.
I'll follow up with you for sure. Thank you.
I think I have the last word, and I know we have to end. And there's so many more wonderful questions in the chat, Kevin, and it's great to see your work. And what I can say is, having talked with you maybe 20 years ago about potential collaborations with psychiatry, it's so great to see all the collaborations you've made and these future collaborations that you're about to make because of your talk today. So thank you so much for coming. And I want to remind people, we're about to start trying to segue to hybrid grand rounds, which means when we have a local faculty who's going to give grand rounds and wants to give it in person, we're going to have them give it in person in our auditorium. And people who want to see live people can come sit in the auditorium, masked and physical distancing still, but to try to actually start to rebuild our community, because I think everyone really enjoyed being together when I had Katrina and our new dean, Katrina Armstrong and Ann Sullivan in person on the stage. And I think it's time for us to sort of start to do that again when we can in an easy way. But with that, Kevin, thank you so much for coming. And Phil, do you want the very last word?
I think Herb would have, knowing Herb, would have loved this every minute of it. And I should thank David Spiegel and Jose Maldonado at Stanford, because I had no clue how we would do this remotely, and they had been trying to figure out how to do it just audio, and they did guide me to change this technique. So it's been an unbelievable collaboration that it's not over. So as you said, this is it's great to be at a place where you can do these things.
Indeed.
And okay, thank you, Kevin, and thank you, Kate.
Thank you, everyone. Bye-bye.
Everybody.